Welcome, everyone. Thank you for coming today, especially on such a beautiful day. My name is Colleen Hazeloop, and I am the Community Programs Coordinator for the Community Relations Department here at Packard Children's. As many of you may know, this year marks Packard's 20th anniversary and a year of complete celebration. Part of the celebration includes today's lecture, The Future in Personalized Healthcare, the Role of Genes, Data, and the Environment, as the fourth and final installment of our 20th anniversary lecture series. The goal of our lecture series is to showcase the technology and innovation here at Packard and to give a glimpse into our future. All of the lectures in this series have been recorded and are available on the hospital website and on Stanford University iTunes. Our anniversary celebration will conclude with our 20th birthday party on Sunday, June 26th. The day will have over 90 interactive booths for children and families, great food from our local restaurants and vendors, a children's performance stage, and of course, birthday cupcakes. There is a save the date invitation on the back table, and we look forward to seeing you and your families there. Now it is my pleasure to introduce today's speaker, Dr. Atul Butte. Dr. Butte is an assistant professor in pediatrics and chief of the new Division of Systems Medicine at Stanford University and the Lucile Packard Children's Hospital. Dr. Butte received his undergraduate degree in computer science from Brown University and worked as a software engineer at Apple and Microsoft before moving to medical school at Brown and training in pediatrics and then pediatric endocrinology at Children's Hospital Boston. After finishing his PhD from MIT, he was recruited to Stanford and LPCH, where he has spent the past 6 years building a lab of 20 scientists, developing new diagnostics, therapeutics, and discovering new mechanisms of disease. Dr. Butte's research has been featured in the New York Times Science Times and the International Herald Tribune in 2008, the Wall Street Journal in 2010, and the San Francisco Mercury News in 2010. Dr. Butte?
Thank you. Thanks for that introduction, and thanks for all of you for coming out today. So it's great to know that I'm the final speaker, actually, for this series. Some of you might have come to the previous speakers, and we've had a great bunch of scientists actually speaking on all sorts of different technologies. And so today I'm going to be maybe changing the tune a little bit. Instead of showing you a lot of different cells and biological experiments, I'm going to stick to a little bit more of the data level and talk about how computer science is going to be changing how we do science. So my usual disclosures, I've started a lot of companies around here. I consult for a lot of companies around here. As the famous Irv Weissman would say, you shouldn't believe another word I'm about to say. But certainly Stanford lets us start companies, and that's what we're supposed to do around here. So we currently live in a data deluge. Some of you might have picked up in The Economist magazine last summer that as a human species, we now generate 2 zettabytes per year. Okay, so we need kilo, mega, giga, tera, peta, exa, zettabytes. Okay, we generate 2 zettabytes per year, and of course that's exponentially growing as well, as shown in the graph here. So next year we'll generate 4 zettabytes. Wired magazine said in 2007, we generate so much data that all of science is just obsolete. Why even ask a question? All the answers are there in the data already, since we essentially measure everything. In fact, the Harvard Business Review last year, in 2010, one of the breakthrough ideas noted this point. Under hacking work, the problem is when a 12-year-old can gather information faster, process it more efficiently, reference more diverse professionals, and get volunteer guidance from better sources than you can at work, how can you pretend to be competitive today? I'll show you what 12-year-olds can actually do in a few slides here. But this is the world we live in today. It's data-driven. Some of you might have noticed my Twitter feed listed on the front page. I just joined Twitter a couple weeks ago, like, I don't know, 500 million other people. And so this current data trend is also there in life science as well, certainly in science. So Harvard Business Review, even more recently, in November 2010, noted that the next big idea, the next scientific revolution is in data mining. That again, we're measuring so many things at a molecular level and clinical level that we should really be taking advantage of all this data that we're capturing. In fact, Nature said data is shameful neglect, that if we don't actually steward the data, actually make it publicly available and transparent, that's actually shameful. In fact, The Lancet earlier this year said it impacts public health if we don't share data. because just because one group published on some data doesn't mean other groups can't find more things in there that are actually going to impact public health. So we've entered this new world of data transparency. Some of you might know when Obama took over, he actually started something called data.gov. There are tens of thousands of data sites at data.gov that people can download and actually start to play with and generate iPhone apps and things like that with just the data that's publicly available. But certainly it's happening in life science as well. A lot of it was actually inspired by this technology here. On the left here is an Affymetrix gene chip. This one chip is the size of your thumbnail, and this one chip actually quantitates every gene in the genome. That means it measures the level of every gene in the genome. And in life science, we get one of these to work. We actually start to make 96 well plates of them. So each one of these is one of these now. Okay, now we'll spend a little bit of time talking about this kind of technology, but this technology measures the blueprints. So we have the DNA that's in the middle of our cells and the proteins, which all the cells want to make to actually conduct business. And in the middle are the blueprints on how to make the proteins. And so this lets us measure all the blueprints on their way to coding for the proteins. Now, Affymetrix and Stanford co-invented this technology. Affymetrix is just down the road in Santa Clara, so it's a really Bay Area invention. This looks unbelievable, okay? This looks just like an Intel chip, right? I mean, this looks just absolutely unbelievable. Would you believe this is a commodity item today already? This is a 15-year-old technology already, okay? Now, what does a commodity item mean? Most people at Stanford don't even care which brand name they're buying. If it's Affymetrix, Agilent, which is even closer than Affymetrix to here, or Illumina, which is across, mostly based in San Diego, but also in Boston. Most people don't even care which company they're ordering from as long as they get the cheaper price, right? That's usually a definition of a commodity. So this chip today costs about $190 to measure every gene in the genome, to quantitate every gene in the genome today. It's an unbelievably low price. So most people just buy these things just based on price because they all essentially have the same features. They're all genome-wide. They're all on a single chip. And so what happened is it's actually not just a Stanford thing that we can get these for $200 each. It's actually across the world. Any investigator around the world can buy these chips for $200 and just essentially measure every gene in the genome. So an investigator studying, let's say, metastatic breast cancer and non-metastatic breast cancer, they can get a bunch of samples. They buy these chips, $200 each, and they can figure out which genes are different. There's more of this gene over there, and there's less of this gene over here, and they can start to figure out which genes tell the difference between metastatic and non-metastatic breast cancer. In fact, you can use this for nearly any condition that's out there. So what happened in the early 2000s is everyone started buying these microarrays, as you can imagine, and everyone started writing papers and started publishing manuscripts about here's a list of genes that are different here, here's a list of genes that are different there. So many people ran these microarrays that the journals said, hold on a second, no more papers on microarrays, okay? So many people are buying these, and it's so commonly used now, this kind of space-age technology, that we don't want to see any more papers on microarrays unless you deposit your data into an international repository. Because we want to make sure other people can double-check all your math and all your numbers and make sure other people can use those samples as well. So whereas most people in science know about these microarrays and they know how to apply them, very few people realize how much data is actually publicly available today because of that effort from the journals. So one of the websites that you can go to is at the National Library of Medicine. It's on NIH campus, and it runs a bunch of these different types of public services, one of which is called the Gene Expression Omnibus. This is the repository for these microarrays. And I can just show you here, circled here, as of a couple days ago, we have 540,000 publicly available samples here, just at this one website, okay? More than 500,000 microarray samples just at that website. But this is just the US repository. We have one in Europe as well. It's also allowed. Here's another 155,000 more samples. There's no overlap. So right now we have about 700,000 publicly available samples today. Now let me put that in perspective. These repositories are certainly growing. The growth of these repositories is finally slowing down to Moore's law, okay? Moore's law is that computational power doubles every 18 months. We certainly know Moore's law in Silicon Valley, okay? Gordon Moore, founder of Intel, noted back in the 60s that computational power was just continuing to double every 18 months. Yeah, we're finally only doubling every 18 months because we had been tripling every year. It's only slowing down because of the next website I'm going to show you, okay? Now, here's the point I really want to plant in your head for a second. In the middle of this website at the National Library of Medicine is a simple search box. A high school kid today, let's say a high school senior, who needs to do a science fair project. She can type breast cancer and download 22,000 samples of breast cancer as easily as she can find a song on iTunes today. Now, if you don't believe me, okay, if you don't believe me that a high school kid can do it, here are the 4 kids my lab has placed in either the Intel, Westinghouse, or Siemens semifinalist or finalist pool just in the past 2 years. Kids who are absolutely enabled to do this today. So these are all Bay Area high school kids who have actually been able to take data from this and actually figure out things that no biologist can figure out. How is it that they can do this? There's something magical about 22,000 samples of breast cancer, right? That's more samples than any breast cancer researcher will ever have in their lab, right? Because by definition, we're going to get anything that any researcher is generating because they got to publish papers on it. We're going to get all their data plus all of their competitors, right? So the person, even the high school kid who knows how to work with the repository in the middle is going to beat any breast cancer researcher. And if it's not breast cancer you're interested in, same story for prostate cancer, colon cancer, or even other diseases as well, not just cancers here. So kids today, by the way, this is a lot more fun than dissecting frogs, right, in high school, right? And this is how fun biology should be when it's digital in this kind of way, and actually pretty applied for medicine and health. So the only reason this repository is kind of slowing down in its growth is because of the next repository, which is based on DNA. So instead of looking at the blueprints, people are now building these strips to actually look directly at the DNA. Some of you know about 23andMe. We'll talk about them in a little bit, where you can go get your own DNA, spots of your own DNA sequenced. And we use those same kinds of chips. Obviously, we don't have to send them to 23andMe. We can use them in our own facilities. And the carrot stick is even harder here. Here, it's not even the journal saying you have to share. Here, NIH says, if we pay you to do this study, you have to share this data, even before it reaches a publication. So here, this is a repository of just DNA samples. And it's a little bit harder here. We don't want high school kids to go download someone's DNA sequence because of privacy and confidentiality. Here, you download the permission form. And you got to get approvals in your own university. Then you eventually get the samples. And here, you got to respect their embargo date. So you're not allowed to publish any paper on this data until after the dates have passed, because they're sharing it open. They're sharing it more earlier than usual. But you can always see most of the embargo dates have already passed by the time you get there. But why is it worth putting up with that hassle? I just want to show you in the middle of this website is the entire Framingham Heart Study. Okay, the Framingham Heart Study is the reason why we know the word cholesterol in the United States. It's a 30-year-old study, longitudinal, of the folks who live in Framingham. It's about 15 miles west of Boston. And it's about 3 generations old now. And yeah, you know, it's sort of interesting to get the DNA in 14,000 people here. But it's more amazing to get 30 years of measurements on these folks. And I can tell you, they never used to let this data out of their hands. They've published hundreds, if not thousands, of papers themselves on this data. But finally, in this era of data transparency, they, too, have to share their data for even a high school kid to now start to study to figure out something they might be missing today. So what an amazing world we're in, that we measure so many molecules in so many samples. Actually, the molecular measurements themselves are commodity items. I can study breast cancer this week. I'll study colon cancer next week, and pancreatic cancer the week after that, right? Because we can actually do this in this kind of way. Now, where we used to get stuck is we used to have a lot of data, but we had a hard time. We could compute on the data. We certainly know how to write programs. Where we used to get stuck is actually convincing then the biologist or the clinician to go test our ideas, okay? Because we're sitting in a dry lab in offices and cubicles. And how do we actually get then back to the lab and convince a biologist or a medical doctor to go try our biomarker, try our diagnostic? What I'm going to show you in the next few slides is even more amazing to me, okay? Just talk about how easy this process is. Now, the other day in the lab, we came up with a marker for pancreatic cancer. Pancreatic cancer is a pretty lethal cancer to get, okay? Usually die within 6 months, only because it's diagnosed late. And it would be great to get a marker for pancreatic cancer. So using some of the publicly available data, we turned our crank. We found some markers that we predicted for pancreatic cancer. And then we wanted to test these actually in pancreatic cancer samples. And so we can go to our cancer samples here. We have a cancer bank here, and we can start to ask around, how many samples of pancreatic cancer do we have? Well, it's a rare disease. We would have to put up some posters, and maybe in a year or two we'd get enough samples here at Stanford to actually be able to test them. How does an informatics person like me find samples? We just Google for it, and we find a company like this. Here's a company selling something called a pancreatic cancer tissue microarray, okay? This is a one glass slide with 100 patients represented. Now, these are all pathological samples that came out of the operating room. Someone stuck a needle in, got a nice circle, and put them on these arrays here. What kind of pancreatic cancer would you like to study today? There are 42 cases of ductal adenocarcinoma, 3 adenosquamous, 1 islet cell carcinoma, and 6 metastatic carcinoma. That means there was cancer somewhere else that came to the pancreas. Then we got some tumors, just some growths, inflammation, hyperplasia, then 20 normal pancreas tissue, and then 10 normals from other normals from autopsies. Duplicate cores per case. So these 101 cases, they got 2 spots on each, okay? This is an amazing sample, 100 different ways to study pancreatic cancer. What am I covering up the price? $395 and literally a little picture of a shopping cart there. You can get this ordered and in your hands to test your ideas in 48 hours today. Okay? Even the samples themselves are not the rate limiting step anymore. How do they get these? This is completely legitimate. It's more legitimate. It's actually acceptable. What happens is there are major hospitals in the middle of the country that take care of patients like this. There's no academic medical center, no researchers interested in studying that disease around there. And that hunk of tissue goes in the trash. I mean, this is going to be a discarded sample. Companies come and say, well, we'll take that tissue before we throw it out. We'll organize it. We're not going to tell anyone who these patients are. In fact, there's no name on this sample. And in the end, they're going to resell that to people like me who can actually make use of that. So the connection between samples and investigators is actually commoditized now. Let's take it to an extreme, okay? Now, I might have ideas. The biologists say, well, it doesn't mean anything until you show it in a mouse model, okay? How do you test this in an actual mouse? Then I show them this website, assaydepot.com, okay? Now, I have no commercial connection to any of these guys, except as a customer. assaydepot.com. What kind of mouse would you like to run today? Would you like to study the bone, cardiovascular, derm, diabetes, genitourinary, infectious disease, inflammation, neurological, oncology, eye, ear, pain, respiratory? What kind of disease would you like to study today? Well, I'm a very part-time endocrinologist. In a previous life, not too long ago, I used to take care of kids with diabetes and growth problems. Here's a mouse model. This one happens to be run in China. Some of them are run in Berkeley. Some are run in Boston. And here's a 16-mouse experiment. And this is a standard model for studying obesity and diabetes. No matter what you do to these mice, they start to overeat. They eventually get diabetes. This is what we've been studying diabetes in for 35 years. And take these 16 mice and do whatever you need to test as a drug for diabetes or intervention for diabetes. Divide up the 16 however you want, 4 groups of 4, 2 groups of 8. And they'll measure the insulin, the glucose, the fasting glucose, all for you. $9,000 for this service, 9-week turnaround time, and literally add to shopping cart. An entire mouse experiment is actually purchasable by e-commerce today. What an amazing world it is for a dry researcher like me today. So now the data's commoditized and the validation methods are commoditized. All you need is someone in the middle to turn this crank like crazy. Let's study breast cancer this week, prostate cancer next week, lung cancer next week. Turn this crank to figure out these diagnostics, get them tested in these preclinical models, and either license them to pharmaceutical companies or start a company around it. And let's just get these things launched, because every step of this way is actually facilitated today. These websites didn't even exist 3 or 4 years ago. Some of you might have read Michael Crichton's book, Next. It's one of his books he wrote before he died, maybe about 7, 8 years ago. Next, it's just titled Next, about what the future of biotech look like. And he actually hypothesized about this kind of thing, where you could just buy samples on the internet. And it's already here. It's not even future. It's already here. It beats throwing these things out in the trash. I mean, there's actually scientific use for this. And we could talk about the ethics and all this during the Q&A, and we got answers for that. So I think the fun part about the science now is that we have so much data, whether it's genetics data or measurements or phenotype, the blueprints, clinical measurements, and these are all the different kind of nifty figures that we generate here. With so much data that I think the fun part is in actually combining it, whether it's the blueprints and the proteins, or whether it's the genetics and the clinical measurements. I think the fun part now comes in actually taking not just one way of looking at these diseases, but multiple ways of looking at these diseases. In fact, I think the only way forward for some of these really complicated diseases like obesity and diabetes is if we really start to combine these data sets. Now, the kids today have a term for this. The kids call this a mashup, okay? When they take a YouTube video of one kind and mix it up with a song of another kind, they call that a mashup. And so what I would say is that what we're doing here are now genomic mashups, okay? We take the data of one, take the data of the other, and actually start to combine these in a kind of nifty way. So genomic mashups, what I'm going to give you stories about over the next few minutes here. In fact, in my lab now, we're so cocky that we think we can study all of human disease from every possible vantage point. We have all of these measurements publicly available. But in reality, my lab lives on a curve. So I'm showing you the 23 people in my lab, including some high school kids. And the way we really work is that we think about one disease from every vantage point, or one vantage point across every disease, and start to make some traction in these directions. So I'm going to give you some funny examples, some anecdotes of what we've been doing in the lab. All of this is either in publication or it's coming out in the next few weeks or months, just to get an earlier kind of idea of where we're going in the lab here. So my hero for the last few years has been this guy Linnaeus. Okay? Linnaeus was born in 1707. He died in 1778. And Linnaeus is famous for one thing. Most people in the audience had to know at one point, kingdom, phylum, class, order, family, genus, species, right? That's called a binomial nomenclature. Kingdom, phylum, class, order, family, genus, species. You all had mnemonics for this, for remembering this, right? Some were clean, some were dirty. You don't have to repeat them here. Everyone had to know this for a test at one point in high school or in elementary school. And what the binomial nomenclature really is to put into a tree all the species that we have. And it's also why we call species by genus and species, Homo sapiens, Mus musculus. And this has been a tree that we've essentially been using for the past 300 years. Now, I was recently in Tokyo, and one of the things you know if you travel to Japan, to Tokyo, is you're jet lagged in the morning. And the thing to do if you're jet lagged in Tokyo is to go see the famous fish market, the Sugi fish market. They're throwing fish at 2 in the morning. It's sort of fun to see that. Right next to the Sugi fish market is actually the Hamariko Garden. It's in the middle of Tokyo. And in the middle of that Hamariko Garden is actually a 300-year-old pine tree. And it was planted a couple years after Linnaeus was born, 1707, 1709 here. And one of the things that hit me about this example of a 300-year-old tree were all the crutches it needs just to keep the branches up and alive. No one expected this tree to make it to 300 years. Every major branch has a crutch just to keep this thing up. And it hit me as an analogy that Linnaeus' tree is the same way today, okay? We can take any species on the planet now, we can sequence its DNA completely, and we can say with all due respect to Linnaeus, this species looks like these guys, but the DNA looks like these guys. So we're going to move it. And we move species all the time in trees, okay? If it's a complicated organism, you get a nice paper in a nice journal. Otherwise, it's a mediocre journal article today to even move a species. It's not a big deal. Now, just for a few clinicians maybe in the audience, I'm just going to say this actually has something to do with medicine too, okay? Some of you might know Pneumocystis carni, PC, that's a PCP. One of the PCP, or pneumocystis, AIDS-related pneumonia, was one of the major sources of mortality morbidity for patients with HIV, with AIDS, going back into the 80s and 90s, pneumocystis pneumonia. And so it's called PCP, pneumocystis carni pneumonia. But in 1999, the CDC, Centers for Disease Control, finally sequenced this and said, oops, this actually isn't pneumocystis carni. We've been calling it that for decades. It was actually pneumocystis jirovecii. It's the wrong name we've been using. So we still call it PCP pneumonia. It's just this P and this C, okay? I can tell you that we still bill the same amount, so you're not going to get a discount if you see a doctor with this. We get reimbursed the same amount. But even a major organism that killed a lot of individuals actually can change after 20 years of using the wrong bug, wrong name. Now, this is a long introduction for Linnaeus, for something that actually everyone forgot about Linnaeus. Even at all the 300-year-old Linnaeus parties we had a couple of years ago. We had a whole bunch of Linnaeus parties a couple of years ago because it was the 300th anniversary of his birth. What did the entire world forget about Linnaeus? Linnaeus was also the first guy to put diseases into a taxonomical tree, okay? And the word for this was actually coined in the 1700s. Nosology means a systematic classification of diseases. And he published in 1763 the Genera Morborum, the genus and species of diseases. In fact, he launched 100 years. The thing to do, if you were a learned physician or learned philosopher, was to come up with your way of categorizing the diseases. And because people had no really great idea of how to do it. Now, this is an example of the kind of kingdom, phylum, class he had for diseases. Let's put the painful diseases here. Let's put the fevers with urinary problems up here. Surprising to me, those aren't painful, but I don't know. Here are the fevers with heavy pulse and topical pain over here. He's not remembered for this because he got it horribly wrong. Why even remember the guy for this? Because he got the species so correct, okay? That's why everyone's forgotten this about Linnaeus. Now let's give the guy some credit here for a second. This is only about 80 or 90 years after Galileo was under house arrest for saying the sun was the center of the solar system. So it's not bad progress for 70, 80 years, right? They're barely doing autopsies. They barely had microscopes, but he made a lot of progress in the idea of even classifying the diseases here. But here was the point that we reached in my lab. We figured if we can now, with confidence, take an organism and sequence its DNA and shuffle those trees around, why can't we do that for diseases as well? Why can't we take some of our trees of diseases and kind of make them correct with molecular measurements, especially those chips I just showed you on the first slide, especially because a lot of them are publicly available, hundreds of thousands of these in these repositories today. Now, you might think that this is actually, this is really easy to laugh at. I mean, Linnaeus, just after Galileo, I mean, he's not going to get anything right. But we actually still use nosology today. Okay, you might be shocked. Where do we use nosologies today? Some of you might know when you go to see a doctor, and if that doctor is still using paper, which the majority of doctors still are, if the doctor is still using paper, when you're done with the doctor, they give you a sheet, and they instruct you, go drop this off at the receptionist's desk, right? And you might stare at that sheet, and you may see a lot of boxes on the back of that sheet, all with the numbers, and the doctor's checked off a couple of these boxes, indicating which diagnoses you have. That's known as the ICD-9 code, the International Classification of Diseases, 9th edition. Okay, 9th edition, sounds old. In fact, in the United States, we're moving to the 10th edition, ICD-10. By the way, just the switch from ICD-9 to ICD-10 is estimated that it will cost healthcare more than the whole Y2K problem we had 11 years ago. Okay, so you can already sense how much of a problem this is going to be to move from ICD-9 to ICD-10. In fact, the 9th and 10th edition of these, it is old. ICD started in the 1860s by William Farr and the Royal College of Physicians, okay? So we're essentially using something, the 9th edition of something that actually started a little after the American Civil War, okay? It's that bad. Now, you might think, there must be something better. Indeed, the pathologists who look under microscopes have a better system called SNOMED, Systematized Nomenclature for Medicine, and it's got hundreds of thousands of disease codes, not just a couple thousand we have in ICD, but even SNOMED is 78 years old today. These are obsolete systems. Let me just show you what ICD-2 looked like, since you've all heard of ICD-9. Here's 100 years ago, Manual of the International List of Causes of Death, 2nd revision, Paris, 1909. This was actually in our library, and you can get all these books from Google Books nowadays today, all for free. And so this book is actually, so by the way, this wasn't a WHO standard, right? Why? Because this predates the United Nations. This was a League of Nations standard. Some of you might remember your European history, okay? So this actually became a League of Nations standard. And it's written the same way our code books are written today. Here, let's use code 39 for cancer of the buccal cavity. That means cheeks. Okay, code 40 is cancer of the stomach and liver, intestines, cancer of the female genital organs, the breast, the skin. And then this book does what we still do in our code books today. We're running out of numbers, enough with the cancer. Just lump all the rest in 45. Okay? In fact, indeed, lung cancer and brain cancer are lumped in 45. In other words, they're saying 100 years ago, it's not even worth distinguishing lung cancer from brain cancer. We don't even have to count them separately 100 years ago. And just to make the point, lung cancer now is the #1 cancer killer. 100 years ago was just cancer, not even worth specifying. Okay? Boy, how times change, even in 100 years. Code 50 was diabetes. It would still be 10 more years from this that Banting and Best would figure out insulin. So we couldn't even tell juvenile from adult diabetes. We could sort of see something's wrong, but couldn't even tell that difference between type 1 and type 2 diabetes. In fact, as an endocrinologist, my entire field didn't exist. If you look up all of my diseases that I take care of, that I'm supposed to take care of, they're all lumped under general diseases. Okay, the general doc had to know about them. All except one, curiously, disease of the thyroid gland. So the thyroid gland sits in the middle of the neck. Some of you might be hyperthyroid or hypothyroid. It's really common to take thyroid pills. Some of you might know if you don't get enough iodine, your thyroid can really grow and grow, causing what's called a goiter. In the US, we get iodine in bread, we get it in salt, we get it everywhere. But you can guess, maybe, that the thyroid can get so big, it can block, I guess it can block your respiratory tube, your trachea. That's why they lumped it under respiratory system. Now, thyroid has nothing to do with how you breathe. It happens to be sitting on the trachea, has otherwise nothing to do with it. What did they know 100 years ago? They lumped it in the respiratory system. They had no idea. Smallpox, cholera, plague, glanders, anthrax, smallest codes in the book. They were absolutely prevalent at the time. I could tell if you had a single case of these today, Homeland Security would be here by the end of the day. Okay, they're all bioterroristic agents today. And amazingly, this international standard has to say, if you run into a patient whose death certificate indicates they died because of a visitation from God, be sure to use code 189, okay? International standard. Now, the same way you're laughing at this, but just imagine how they're going to be laughing at us 20 years from now. Why even 50, right? Those silly fools didn't know this arthritis and this bowel disease were the same thing, right? You can imagine how they're going to be laughing at us because we lumped these things completely arbitrarily today. So we said, let's just throw this whole thing out and just start to build a new one with all the different data sets we have. Indeed, we published in a lot of different papers. It's a student who's actually building this the most, Joel Dudley, in my lab. We now can build a huge grid of 300 diseases this way, 20,000 genes this way, and we know how much every gene changes in every disease, all from the public measurements. Okay, we don't even have to run our own experiment. We add about a disease or two every week. That's how many people have been running these arrays. So we build these huge tables. Here's leprosy. Here's Marfan syndrome, what Abraham Lincoln probably had. Here's the left ventricular hypertrophy. Here's salmonella. We got all the measurements, all the diseases, and now we can start to compare one disease to another. In fact, let's just compare all the diseases with each other and make a tree, right? We started this exercise to build a tree. And here's a tree. It doesn't look like a tree you're used to. There's a root up here somewhere, and there's branches down here. And the idea here is that the branches towards the bottom are more similar to each other than the branches all the way up at the top. And I'll walk you through this because it's impossible to read something sideways, but I'm going to walk you through this so you can think about what a gene expression or a nosology using molecules looks like. Over here, we saw an interesting branch between colon cancer and colon polyps. I could tell many of you in the audience are already over age 50. You've already had a colonoscopy because that's standard preventative care now, because colon cancer is a killer in the United States, that if you're over age 50, you're going to get one, if not every couple of years, you're going to get a colonoscopy. And if they see a polyp, they stick a needle and they biopsy it to make sure it's not cancer. So it's sort of interesting. We sort of already knew that colon cancer and colon polyps look like each other. Over here, we had cervical cancer. Actually, we had 4 worsening stages of cervical cancer. And we found that as cervical cancer worsens, it approaches this incredibly rare pediatric disease called APOSED. Now, APOSED is a weird disease. It's incredibly rare. What it is sometimes kids are born having autoimmune problems. They don't know, their body can't recognize what's part of themselves and not, and they start to wipe out a lot of their organs, including a lot of their glands. So it's a pretty rare disease, kind of interesting disease. But why is this interesting? Because this cancer, cervical cancer, is more similar to this pediatric disease than this other cancer, colon cancer. Why actually did we even think all the cancers look like each other? Just because the same kind of doctor takes care of them. You realize how silly it is actually when you think about it. That at a molecular level, we're going to be mixing adult disease and pediatric diseases quite a bit. Now that actually might help us. And what got us written up in the New York Times a couple years ago was a tiny branch over here that was similar. You see, heart attack, myocardial infarction, everyone knows what a heart attack is actually similar to muscular dystrophy. Muscular dystrophy is a rare pediatric disease. Kids are born having a muscle weakness, and they eventually they can die from respiratory problems or heart problems. There's a famous telethon every year to raise money for muscular dystrophy awareness and for research. Why is that an interesting pairing? Why is it interesting? Because today we have 40 FDA-approved drugs to treat heart attacks, and we have a whopping 0 to treat muscular dystrophy. And now I know having a heart attack is really similar to having muscular dystrophy. Why can't I take one of those drugs that we use to treat heart attacks and try it in a kid with muscular dystrophy? Even before I put it in a kid. If it only cost me a couple of $1,000s to try it in a mouse model of this disease, why don't I take some of these drugs from these diseases we're really good at treating and try those drugs in diseases that we have no idea how to treat today? So the idea is called drug repositioning, to find a new use for an existing drug. Drug repositioning goes way back. The earliest one I can think of is an interesting cardiac drug, cardiac trial for angina, that had this interesting side effect of hair growth, which today is the minoxidil ointment we use. Minoxidil started as an angina trial, okay, with an interesting side effect. But of course, the famous drug everyone knows about repositioning was another interesting cardiac drug. And nobody wanted to return this drug after the trial, which today is now known as Viagra, also started in a cardiac trial. Now, very few people know that Viagra has done this again. We have another drug, same chemical, but different drug called Revatio. Revatio is used for what's called pulmonary hypertension. When the blood vessels from the heart to the lungs get constricted, Viagra or Revatio is good at opening those up as well. So Viagra is actually switched twice into new indications. And you know what? Instead of finding these things by accident, why don't we actually start to look for these using the public data and these kinds of computational tools to figure out diseases are like each other? So fast forward, I'll show you some interesting stories. In the lab, we predicted, using the same approach I just basically hinted at, we figured out a seizure drug that we thought should work on Crohn's disease. Now, some of you might know about Crohn's disease, ulcerative colitis. It's a pretty bad inflammatory bowel disease. You basically there's constant chronic inflammation in your bowels. And we predicted a seizure drug should work on that, okay? No one's ever shown anything like that. And just to give you an example, here's an example how you would do this. A couple $1,000 gets you these experiments once we figure out the computational side. You take rats, and you can do a rat study. You inject a chemical called TNBS. It makes them get the Crohn's disease. And you could treat them with something we know works, like prednisone. It barely budges. You try topiramate, Topamax, that's a seizure drug. And you can see on both tables, whether it's the damage or the gross pathology, you can treat them almost back to, on the way to actually not even getting the disease. So here's just a regular rat. Here's rat with the Crohn's disease. This is what happens when you treat with steroids. Yeah, it reduces the inflammation, but also inhibits wound healing. Here's the seizure drug. It looks right back to the normal. And in fact, we can now, because, again, just tell you a kind of funny story. When we wanted to do this experiment, we put out an RFP, a request for proposals. We found 100 companies in the United States and all over the world that would just do this experiment for us, okay? Why did we pick the one out of the 100 near Boston? Because they could do a rat colonoscopy, okay? Need I say more? You know how big a colonoscope is. They got such a tiny one to do it in a rat, so they can take pictures of these rats even while they're all still alive. And so we have these gorgeous pictures of just as a normal rat, this is a rat with the Crohn's disease. With prednisone, it's a little bit better. With the seizure drug, it's back to normal. So we convincingly did this 3 different experiments, now 3 different labs. This drug works on Crohn's disease. Now we said, let's try it again, okay? We found an ulcer drug should work on lung cancer, of all things, okay? Now what's neat about this ulcer drug? This is over-the-counter, okay? It's called Tagamet. It's otherwise known as cimetidine. Tagamet is over-the-counter ulcer drug. You'll see in the ads for it. It's actually available in generic form. So we figured Tagamet should work on lung cancer. So how do you do this one? What you do is you take lung cancer samples from humans, lung cancer cell lines, inject them into a mouse, and then you watch them as they grow. This is how science is done. You might like mice or rats, but this is how science is done. You watch them 11 days. If you do nothing to these mice, the tumors triple in size in just 11 days. Pretty bad cancer, yeah, lung cancer, right? We don't have a lot of therapies. If you give them an anti-cancer drug, doxorubicin, it only doubles in size, okay? It's still growing like crazy after 11 days. The 3 in the middle, the slower growth, are actually the cimetidine. In fact, this is actually 25, 50, and 100 milligrams per kilogram body weight. In fact, they're in dose-dependent order. So that proves to us that this drug actually works. Cimetidine works on lung cancer. The highest dose we tried here is actually the kids' dose, the pediatric dose. We didn't even try the adult dose of this medication yet. I think we have a long way to go to actually get cimetidine to work in lung cancer. Now, the challenge to us, in fact, we've done this now a couple times. We know this drug works as well. There's all sorts of interesting challenges. How do you actually get a drug like this to patients? Yeah, it's nice to show and cure cancer in a mouse, right? But how do we get this into patients? This is a really tough story because this is already available in generic form. So in other words, no pharmaceutical company today is going to want to pay for a trial here because there's no money to be made at the end of the day, even if this drug is successful. So now we have to be even more creative, right? Let's come up with something that's unique about cimetidine that there's no generic form available so that the pharmacist at CVS just doesn't switch out the drug. So how else can we think about giving an ulcer drug to lung cancer? Maybe I can just make an inhaler for an ulcer drug. Maybe I can come with an enema form of the seizure drug. We can try different forms of the drug and then maybe add value and actually get enough value into that to actually get the trial done. And this is the process that we're in now. And you had another startup company spinning out of this just in the Palo Alto area now, taking this approach and moving it forward. So just to make this clear, using the same data a high school kid can get to today, we can now find 2 new drugs, this just in the past 2 years, one for lung cancer and one for Crohn's disease. So that's on the therapeutic side. Let's go a little bit faster and talk about the diagnostic side. You know, one thing that we do really well at Packard Children's Hospital is transplanting organs, especially into kids. We do transplants of the liver, heart transplants. Heart transplants is a tough procedure. We have some of the world's best here. We do kidney transplants. We're a center for excellence for kidney transplants. And one of the things that goes wrong after kidney transplants is or heart transplants, one of the things that goes wrong is the kid wakes up or the adult wakes up and all of a sudden the body says, this isn't part of me, okay? I need to get rid of this organ. It's called acute rejection. And now one of the problems with acute rejection is that it's not painful. The patient doesn't even know it's happening. So the only way to tell this is happening is actually we could take ultrasounds and look at the organs. Maybe we get a little bit of a hint. If we get a little bit of a hint that something seems fishy, we stick a long needle in, and then we hope that we got a sample that's actually accurate, and then look under the microscope, and maybe we're going to see that there's acute rejection going on. Sticking in a long needle in there doesn't seem particularly nice to me. And so we in the computation and informatics side said, we need to come up with a blood test for this thing, okay? And we can be so naive in informatics. We said we want one blood test no matter which organ went in, heart, liver, kidney. If it's rejecting, we want the same blood test to go up, a universal blood test for acute rejection. So how do we find that? Well, publicly available data again. We can get from those same repositories kids that have already had a kidney with and without rejection, adults who have gotten a kidney with and without rejection, and even kids and adults that got a heart transplant with and without rejection, publicly available today, okay? In other words, a high school kid can go download this data. In fact, the last kid I just showed you got the the Westinghouse and the Siemens finalist actually did this. We started this with 3. He found 35 transplanted organ experiments like this and intersected those. Again, a high school kid actually beat our own lab, you know, because we only found 3, he found 30, okay? But even with 3, you can see 2,000 genes up here, 2,000 genes up here, 200 up here. You can't chase down 200 different candidates. But a very simple Venn diagram, right? Even my 2nd grade daughter actually learned about Venn diagrams already, right? 45 in common between this circle, this circle, and this circle. And 45 is a number we can start to chase down. In fact, we said, let's go chase all 45 down. And instead of looking at the blueprints, let's just pretend that there's actually a protein on the other end. So we're going to look in the blood for the proteins for these 45. Now, when you look at proteins, there's a technology called ELISAs, enzyme-linked immunosorbent assays. This is 1970s protein technology, okay? We're not so fancy here. These ELISA kits are about $500 each. And we said out of the 45, 10 of them were available. Literally, you could buy a kit for these 10. The one thing I learned in my 1 year of spending time in a biology lab, if there's a kit, go buy the kit. Because trying to make this stuff yourself, you're always going to make it wrong. So we said, let's just pull the trigger on 10 of the 45 that we could buy a kit for. And let's try all 10 in brand new samples of acute rejection stable for kids with kidney transplant. Simply asking, can we get any of the 10 to work? Of the 10, 3 were brand new blood tests for acute rejection. This is what we call a sensitivity specificity curve for those of you who know in the sciences. And we got decent performance in sensitivity and specificity for 3 out of the 10. We take the better one of these, a gene called PCAM1, and we said, let's go downstairs to the basement to the pathologist and see what exactly does it look like. And here's kidney after transplant with rejection, without rejection. And it's stained for PCAM1, one of the markers. And you can even buy eye. You don't even need to be a pathologist. See, there's more here than here. Okay, so we think that it's working. And it's in the kidney that's actually being rejected. But you know what? While we're downstairs in pathology, let's just go look at the other organs, the liver and the heart. And the same markers up in the liver and the heart as well, just like we predicted. Now this, just showing it up in the heart here, helped us convince our heart transplant collaborators, because heart transplants are rare still, even at Packard Children's. And when they get that blood and serum, they save it because it's extremely precious to go thaw that out and actually test these markers. This was enough to convince them to thaw out those markers to test our 3 new tests, and 3 out of the 3 were also up in heart transplant rejection as well. So now we got 3 brand new blood tests for rejection of the heart and kidney found using the same data a high school kid can get to. Where are we going with this? Turns out 2 of them you can find in the urine. Okay, so what does that mean? Instead of saying, yeah, you're rejecting your organ 4 months from now when you actually make it to clinic, why don't we just come up with a pee strip that kids or adults can just pee every morning on the strip and say, you're rejecting today. Go up on your meds today. Let's get that rejection under control today. Not when you come back in 4 months to a clinic, and because you otherwise don't know that this is happening. Let's take care of it today. So where we're going now is a pee strip for rejection today. It's something that any kid or adult can actually use. Using public data, we can get to all of this today. It's an amazing world. So now let me end with the personalized medicine part of it, okay? And even more technology here. Some of you know that we have 23andMe, which is right down the road. We have this idea that we can get our DNA sequence. You know, in 2003, the Genome Project finished, and we now know what the Human Genome Project is. So the easiest way to think about the Human Genome Project is it's about 300 copies of the joy of cooking, okay? You all, essentially everyone knows the joy of cooking. Many of you buy it, and you know how thick it is. It's about 300 copies of it, okay? It's a huge amount of recipes, because all the DNA are recipes to make proteins, okay? And there are subtle differences. Imagine the joy of cooking, but 1 in 1,000 of the letters are different between any 2 of you, or 2 of us, okay? So that's how many differences there are in our DNA, about 1 in 1,000. So many companies working on actually sequencing the DNA since the Genome Project was done, one of which is called Helicos, and it's actually started by Stanford professor Stephen Quake. Steve Quake is a remarkable individual. He's already full professor here. He's about my age. He's already started 2 or 3 publicly traded companies. He's a great entrepreneur. One of the companies he started is a company called Helicos. It's out in Cambridge, Massachusetts. And he actually paid $35,000 2 years ago to just get his genome sequenced. Okay? And he put it out into a journal article in Nature Biotechnology. It's a good journal. And he put his whole sequence out there, publicly available. First professor to get sequenced, blah, 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 2009. Okay? What was left unanswered was, what's the medical use of having your genome sequenced? Okay? Yeah, this is neat. This is fun. Maybe we can figure out our ancestry. What's the medical use of getting your genome sequenced? And that we answered actually last year. It's not even been a year now. Well, actually, today is exactly a year. Paper where Steve Quake, Russ Altman, Hank Greeley, George Church, you and Ashley, myself, and all of our teams got together and figured out what is the clinical use of having your genome sequenced. And actually, this article was picked up in about 300 newspapers. This is, we like this figure the best. This is actually translated into Czech. They took our figures, and you're going to see, actually, it was translated all over the world, the impact of this paper. This came out of Stanford and Packard Children's Hospital, all of this work. And I'm going to walk you through what we did here. First, let me present Stephen Quake to you as a patient, because what happened is he presented as a patient, not just a faculty member. He came in as a patient. Here's a 40-year-old male who presents to his doctor in good health with his entire genome, okay? He has no symptoms. He exercises regularly. He takes no medications. Why did he come to the doctor? Because his nephew had recently died. His teenage nephew had recently died from sudden death, okay? Tragedy, okay? Died in his sleep, nephew. Healthy kid. And Steve Quake goes to his cardiologist, you and Ashley, who's on staff here at Stanford, and just simply asks, am I at risk for sudden death? Here's my genome, doc, okay? Patient has a heart rate, blood pressure, blah, blah, blah. He presents with 2.8 million differences, SNPs. These are single base pair differences in his genome compared to everyone else. 752 copy number variants. That means he's got an extra or missing copy of a chunk of his chromosome. Everyone else has that too. He's got 700 of these. And by the way, doctor, your next patient is here, and you're already 15 minutes late to see them, okay? How are we going to deal with the physician of the future to give advice on a genome in the scope of a 15-minute encounter? That was our exercise last summer, okay? Let's show you how we did it. What are we going to get out of our genome? Okay, so here's his family history here. Here's the patient himself, Steve Quake. And here's the 19-year-old. It's actually his nephew, died of a sudden death. And so first question we have to ask is, what exactly is the risk? So one of the things we learned actually from sequencing Steve Quake, and we actually learn now in the subsequent year, is it turns out we think we knew where all the differences were in the DNA, but every time we sequence someone, we're finding new differences that we're seeing for the first time in that individual, what we're calling private variants. They're not really private, it's just that we haven't sequenced enough people in the world to really know where are all the differences. So what that means is, by definition, if we've never seen those differences before, we really don't have a bloody clue what it means for anyone, because we've never studied that difference, right? Turns out Steve Quake has a bunch of them, even in the genes and the proteins that help his heart contract, okay? And if it helps your heart contract, it can actually maybe mess up your heart. So he actually has a couple of these. We told him, everyone knows, he's disclosed this to everyone, so I'm not sharing any private information here, but there are some genes that actually have these rare variants that we had never seen in anyone else. So what do we tell Steve Quake? We said, instead of the extreme exercise that you're good at doing, perhaps you should consider moderate exercise. Now, if that doesn't sound like useful advice, you're probably right, okay? To go from extreme exercise to just moderate exercise doesn't mean very much. He, of course, hasn't taken our advice. We've yet to figure out which gene in the genome is responsible for compliance in patients. We have non-compliant patients all the time. He's another one of them. So that's the heart story. We can make a lot of predictions now on the drug side. So this is a field called pharmacogenomics. This is done by Russ Altman and his team. I'm just taking his slide. And for 100 different drugs, there are only 10 here on this list, we can make some kind of comment. So for example, well, Steve Quake, you're not at high risk of myopathy. It's a bad muscle disease. 1 in 30,000 patients are on statins. I'd venture to say a lot of you are on statins. 1 in 30,000 people actually have a high risk of actually could be fatal myopathy. He does not have that risk. He's likely to not need as much of warfarin, a blood thinner, as other people might. He's likely to respond well to fluvastatin, maybe to pravastatin. But if he gets diabetes, he's not likely to respond well to metformin and troglycazone. So now for 100 different drugs, we can start to predict, is he going to get a side effect? Is he likely to respond well to this? And how do we have to adjust the dose for him? Okay? 100 drugs, we can do that today with this sequence. So my lab was actually tasked with all the rest of medicine, okay? There's cardiac, here are the drugs, now here's the rest of medicine, okay? And so long story short, for the past 4 years, we knew this world was coming to this point. We had built this initiative in my lab to just basically go read and computerize every paper that's ever been published in the last 10 years that's ever linked any spot in the DNA to any disease, okay? We built this huge database. And how do we build it in my lab? I already showed you we work like a business. We outsource the whole damn thing, okay? Because we don't have the space or the people on campus here to do this. Every one of my folks now is a manager, and they're managing a team that could be elsewhere in the United States, it could be anywhere in the world, to actually get this work done and then show how we can use it on patients. This is what we've done. This is Alzheimer's disease. Steve Quake, as a 40-year-old white male, has a 9% chance of Alzheimer's disease. That's without knowing his genome. Now let's start to add back all of these spots in his DNA, okay? And now I've ordered these spots in his DNA from the most believable to the least believable. What do I mean by that? Here's a spot in the DNA that's been published in 6 papers having to do with Alzheimer's disease. This one's in 5 papers. These are in 4 papers. And you know, if they're in the same number of papers, we've ordered them based on how many people were studied in those studies, okay? So this was in 4 papers studying 6,000 people. This was in 4 papers studying 4,000 people. These were in 3 papers, 2 papers. These were only in 1 paper. This one's only in 1 paper in just 170 people studied. So now let's pretend I'm a gray-haired doc, and you're a patient, you come to me, I'm a gray-haired doc, I've seen the world. I don't believe anything in genetics if it's not in more than 3 papers, okay? 3 or more. So let's say I draw my line here. I say, well, Steve Quake, you started with a 9% risk. Well, I can show you now we've dropped to about 3.65% chance of getting Alzheimer's disease. Now let's pretend I'm a young whippersnapper doc. I believe everything in the literature, even if it's in medical literature. And I go all the way down to the bottom and say, well, Steve Quake, you have only a 1.37 chance of Alzheimer's disease. And now it's going to be up to you to decide, what kind of doc do you like working with? You know, do you like a younger doc? Do you like an older doc? People have preferences here. I'm not going to be the one to tell you these are the believable ones and these are not. This is the data, and we can start to figure out together. Now, both of these numbers are lower than the first number, so that's good. He's probably protected for that. But if we can do this for one disease, let's just do it for all of them. And here's 35 of the top 1,000 diseases we had. Here's Alzheimer's disease. The black triangles indicate what we thought the risk was before we did the genome. And now the lines indicate how that's shifted after we've done the genome. So yeah, here's the one big one he's protected for. But these, he's actually at higher risk: obesity, coronary artery disease, type 2 diabetes. So those are probably up there for a lot of us around the world. And maybe, even if I can't fine-tune the exact percentages of risk, maybe if I could put you into one of three buckets. Look, we're all going to die in this room today, right? We're all going to die someday. Is it cardiovascular, is it cancer, or is it neurological? Because even one of those three buckets might actually help us figure out what kind of prevention we want to do to actually control for that. Absolutely. So he's at higher risk here. But you actually bring up a great point about depression. Your DNA does not govern your destiny, okay? It sets up a risk. It's the environment that's actually going to pull the trigger of a loaded gun here, okay? My colleague Paul Wise, he's a learned guy, he's a faculty member here at Packard Children's Hospital, he has a great saying. In a famine, nobody's obese, right? The genes are still there, right? They didn't go away. But if you're not able to eat more, nobody's obese in a famine, right? That's an extreme example. The genes do not govern your destiny. In fact, as you're going to see in a moment, I think it's mostly the environment that governs our destiny more than anything here. So we can say some bad news of some of these, some good news for these others. But it cannot just be about making this prediction and giving bad news and good news. I think the future of the 15-minute genomic encounter is for the doctor to give you what should you do to compensate for your genome, right? We're a long way away of fixing our own DNA, okay? I can tell you that for certainty, okay? But what should I do to compensate for what my DNA looks like today? And so in the end, we came up with this kind of nifty figure. This is the same diseases now, but now showed with different font sizes, okay? Bigger font means really high risk. Smaller font means less risk. Tiny font means even less risk. And so the first thing we're going to note is diseases don't exist by themselves, right? One disease can cause another disease. For example, by itself, Steve Quake is not at high risk for hypertension, high blood pressure. But he's at high risk for obesity and type 2 diabetes, both of which can cause hypertension, right? So this is a graphic to show him if you get any one of these, don't skip the blood pressure screening because you still might get this, okay? The other disease might cause it. All around the edge are all the known environmental factors that also cause these diseases, okay? And that's what we should be targeting. Smoking, exercise, diet, and alcohol. Yeah, boy, I didn't need a genome to tell me that I shouldn't smoke, I should exercise, should watch my diet, limit my alcohol. Probably any of us who gets our genomes, those are going to be our top 4, right? But all in tiny font here is pesticides. Yeah, Steve Quake, I know you like to garden. Stay away from the pesticides. Go hire a gardener. Because you're at slightly higher risk for Parkinson's disease. Go hire a gardener. Don't touch the pesticides, right? This is extremely crude. This is extremely first pass. But this, to us, is a kind of environmental prescription that a doc can give to a patient informed by the genome. You can imagine a doc saying, well, Steve, you're doing great with smoking. You're not a smoker. You're exercising well. Let's focus on your diet for the next 6 months. Come back to clinic in 6 months. Come back to my clinic practice. Let's see how you're doing. If you're doing well there, then let's work on the alcohol next, right? And you can imagine how a doctor and patient talk like that. But now advice given based on a genome. It is still a testable question. We don't know the answer. If I tell you to lose weight, and then I tell you to lose weight because I have your genome here, are you more likely to lose that weight? We don't know the answer to that. It's a testable hypothesis. We just don't know. You can bet we're going to figure that out over the next few years, that if you get the advice with your genome in hand, whether you're more likely to be compliant or not, we don't know. But you can see that's a kind of interesting question to ask. Now, before you start to think this is unbelievably pie in the sky in the future, let me just show you what's happening in the Bay Area. We had companies, 454, which is a high-throughput sequencing company purchased by Roche, Life Technologies. We used to call those next-generation sequencing. It's already previous-generation sequencing. These two are already in the Bay Area, okay? Let me just show you 2 companies, one 5 miles north of Stanford, one 5 miles south of Stanford. Pacific Biosystems is in Menlo Park. I think they're just right off on Willow. They're on track to come up with technology to sequence the entire human genome in 15 minutes. Okay? In fact, this is old news. Discover magazine noticed this 2 years ago and came up with this article, the Jiffy Lube of the genome. Okay? Now, some of you know Jiffy Lube can change your oil supposedly in 30 minutes, and this is faster than that. Okay? The entire genome, runtime in minutes, cost of hundreds of dollars. I talked to my friend there. They generate 20 terabytes every 15 minutes. Okay, you talk about the data that's being generated, 20 terabytes every 15 minutes. It is no mystery to all the IT companies in our area that life science is a great customer today, okay? It's unbelievable how many hard drives you need to sell to folks like us, okay? That's in Menlo Park. Let's go to Complete Genomics. They're in Mountain View, just right next door to Google. They already have announced they sequenced 500 human genomes last quarter. In fact, they are now sequencing 500 genomes every month. Now, let me put that in perspective for a second. In 2003, we finished the Human Genome Project. It took 13 years and $3 billion. Today's price, that's already a little old. Today's price is $4,500, okay? So we dropped from $3 billion to $4,500 in about 8 years. Nothing ever in IT dropped like this. This completely demolishes Moore's Law. And there are many graphics by the Genome Institute at NIH, many others that just plot Moore's Law and how we're just plunging compared to Moore's Law here in terms of price per base pair. Illumina is down further down in San Diego. They're now down to 4,500. We have Ion Torrent. A lot of these have been written up in the New York Times. You might just pick up some articles in the past few months. In the Science Times, you can see a lot of these companies mentioned because these are unbelievably hot companies. Indeed, they're being sequenced all over the world. This is an article from Nature a couple months ago. In the United States, every one of these blue guys represents a genome we had finished, 500 genomes finished in October 2010. The purple genomes indicate 500 genomes completed by 2011, end of this year. So you can see how many we did last year, how many we're going to do this year in the United States. But this is Beijing genomics. If we ever needed a Sputnik moment, here's one of them. Last year, you couldn't even buy a sequencer from Illumina because they bought them all in Beijing. Okay? This is not a US-only phenomenon. The entire world has figured out that the future is in DNA sequencing. It's European as well. We have to get with the program. Okay? This is exactly where it's going. Other companies, other countries are investing heavily in genetics today. Okay? So they're on track to now sequence more patients than we are in the United States. But in all, 30,000 people will get to genome sequenced this year. 30,000 just this year. Now, let's skip ahead and think about the environment for a second, because especially about the question. Genes can load the gun, but it's going to be the environment that pulls the trigger. Okay? And I just want to make sure you leave with that point, because the genes don't govern your destiny. What do we thought, when you think about the environment, what are we talking about? Well, these are just articles in the past year about where we're learning about the environment causing problems. BPA, bisphenol A, is constantly in the news. Now we just learned they're in Coke cans. We knew they were in canned goods. We obviously knew they were in the Nalgene bottles we use to drink, when we're jogging. Well, now we know they're in Coke cans. This was a couple days ago, so now they got to get banned from there. Phthalates, this is just from Time and New York Times. Arsenic and uranium in our drinking water. Pesticides on a lot of different foods that we eat and drink. And you start to wonder, do any of these play a role with disease? People are actually interested in studying the role of the environment with disease. I mean, it's been studied for 50 years by the epidemiologists. But the idea here is the environment should have an analog like the genome, okay? If all the genes make up the genome, maybe the entire environment makes up the envirome, okay? We need an ohm word for this. We love making ohm words in science, okay? So maybe we need an exposome or an envirome with all the different exposures that one can get: radiation, stress, lifestyle, obviously chemicals, pollution. And people are starting to think about how do we study the envirome and the exposome. So these are slides from my graduate student, Chirag Patel. Again, these are just fresh graduate students who are able to study the environment and the exposome. Now, some people have actually taken upon themselves to figure out their exposome. So this is an example of a website of what's called the Human Toxome Project, where people have just actually gotten their own environmental measurements done and actually made them publicly available so people can actually start to study this. You can get a listing of all your body burden. It's called body burden assay, where you can get all the chemicals and environmental factors by sending in your blood and urine to companies and get what's called body burden assay. And they look at all these different kinds of chemicals, DDT, lead. But if you're really interested in this, the book I'd recommend the most is this book called Experimental Man by David Duncan. If you're really into this kind of genome, medicine, environment, David Duncan is a New York Times science writer, but now he's a faculty member at Berkeley. And this guy has actually done everything, one of everything to himself, okay? He's gotten every genome done, 23andMe, Navigenics. He's gotten every imaging study done. He's actually gotten his whole environmental factors done. And actually, he's made them all publicly available. So you can go to his website and just download all of these measurements on him, okay? Kind of cool high school kid project, you can imagine. And so one of the kind of cool stories from this paper, from this book actually he tells, is that he got his genome done, and he has these nifty graphs, just like I showed you. But some of the companies say he's at higher risk for this and lower risk of that, and he's not really clear where he's going. But actually, he was born in Kansas City. He was born and raised in Kansas City next to a lake. Okay? He's a little bit older than me, maybe 40s, 50s. He still has a massive peak of DDT in his body. Some of you might know we banned that sucker decades ago. It's still in our bodies. Okay? But the only peak he has even higher than that is flame retardant. Okay? Because every piece of furniture in California has flame retardant. Okay? Every airplane seat we sit on has flame retardant on it. Okay? So he's got two big peaks here. He's thinking, yeah, genes do this and that and left and right. But he has to think the flame retardant and the DDT's got to do something. So why don't we study that? Why don't we study the role of the environment? Because it's not sexy. This is the problem. We have these nifty chips I just showed you on the first slide to look at the entire genome. We don't yet have chips to study the environment. If I had to start up yet another company, I would be on that. We need environmental chips to think about how to study the environment in blood and urine so that we can start to use that in research. But until then, we said, can we use public data on the internet to help us kind of simulate this? And what we did is we went to a survey called the NHANES study, National Health and Nutritional Survey. So the CDC, Centers for Disease Control, run what's called NHANES. Now, any of you who have kids know when you go to see your pediatrician for the kid, they plot the kid on the height and weight on the growth curve, right? If you stare at that growth curve long enough, you see in small lettering that the measurements come from the NHANES study. This is a 52-year-old study where all the heights and weights of American kids and adults are measured. They're sampled and measured, and that's where we get our growth charts. But NHANES has done much more than that. Actually, NHANES is the reason why we don't have leaded gasoline anymore in the United States. Because decades ago, they figured out a connection between mental deficiencies and leaded gasoline and just lead in the environment. So it's been going on for 50 years. But besides the height and the weight measurements, what's really neat is that they've been measuring in blood and urine tests on hundreds and thousands of Americans, okay, for environmental toxins. It's a complicated slide. I'm not going to walk you through all of it. But you can see in 4 different cohorts, 1990s, 2001, 2003, 2005, they've measured 13 different heavy metals, PCBs, phthalates, perchlorates, pesticides. All of this data, believe it or not, available on the internet. So Chirag Patel in my lab said, let's just go test every one of these factors and see if it has anything to do with type 2 diabetes, the public health menace in the world. Right? Yeah, and we know diet and exercise are the reason why kids are getting obese and they're not exercising. But are we so sure there's no other environmental factor that's causing the diabetes epidemic today? And lo and behold, when we tested all these environmental factors and plotted it just like we plotted genes, the whole simple approach here, the reason why this was written up in 100 newspapers is because we started to think about the environment like the genes. Geneticists today, if they want to go find a gene, they don't say, let's guess it's this one, let's guess it's that one. They say, let's just study them all and just find it, right? But why can't we use that approach in the environment? We said, let's just go study them all and figure out something new here. And lo and behold, it's a complicated slide, but I'll just tell you the #1 factor for type 2 diabetes we found was heptachlor epoxide. It's a derivative of heptachlor, which is also a banned pesticide, banned in the 1985, 1986 because of carcinogenesis. Believe it or not, it's still legal to use heptachlor. Why? Because in some instances, like when PG&E has an underground transformer and it's infested with fire ants, believe it or not, you're still allowed to use this random chemical. Okay, so it's banned, but still actually used in our environment today. When I mean our environment, I mean our environment. This is a pesticide. Pesticides are used in the Central Valley. The water comes here, so don't think we're immune to this stuff. Don't think environment means, the poor side of town. This is us too. And in 2 independent cohorts of Americans, heptachloroperoxide was significantly associated with type 2 diabetes. It's a brand new factor here. We only found it because we started to think about the environment like we think about the genes. Let's just look at them all. And in the meantime, let's work with companies, and there's researchers at Stanford, Berkeley now that are coming up with sexy chips to study the environment now because we need that. The genes are not going to be enough to write your destiny here. So let me just end with a kind of couple interesting thoughts. We all know that this whole approach is called personalized medicine, right? Nobody wants to be treated like everyone else. And so based on your DNA, maybe based on your environment, maybe your cancer might look different than someone else's cancer. We have this idea of personalized medicine. We should personalize your care compared to someone else's. The big problem, though, is this, okay? Colliding directives here. We're all also aware that we need to improve the quality of our care. You all read about in newspapers all the time that we're making mistakes in hospitals, that the wrong thing comes out, the operation's done on the wrong side of the patient, that we're overdosing patients. At Packard, we're particularly good at finding and making sure we don't do these things, one of the best in the country. But what the problem is how do we treat every patient differently at the same time maintaining quality in all the patients, right? Famous examples is like Southwest Airlines, right? They only fly still one kind of airplane, right? 737s, if I remember correctly, right? They maintain quality. That way it's completely consistent how they take care of their passengers here. How do we treat everyone safely while treating everyone specially, right? How are we going to do that? So what we've done is we've created a new division to think about this, and that's this new division of systems medicine. Think about a kid or a patient with a chronic problem. Here's a kid right now going towards a good outcome. But right here, as we're seeing them, they're heading to a poor outcome. And what we realized is everyone in the world is studying what in the genome and the genetics is going to change this kid from not ending up with a good outcome with this disease but heading towards a poor outcome. But even if we have the entire genome sequenced, if this kid or family can't afford the meds, if they're exposed to pesticides, it's all for naught, because all of that is going to alter whether this kid heads towards a poor outcome instead of a good outcome. But even still, if we have the genome sequence on every patient and we got them all affording their meds and in good environment, if we as doctors can't practice quality care, that also will help them switch. Instead of a good outcome, will make them go towards a poor outcome. The quality folks, the geneticists, and the social epidemiologists have to all work together. I can tell you no other place does this except for Packard Children's Hospital now. And the idea is every single patient we should learn from and apply it back to every patient so we work earlier and earlier, figuring out which of these kids and adults are going to get these chronic diseases and making that intervention as early as possible. And that's what we call data-driven systems medicine. So just to skip ahead, take-home points. To me, personalized medicine is great. We're going to get the DNA. It's $4,500 now. I can guarantee you by the end of this year, next year, it'll be $1,000 to sequence your genome. It's not a big deal, okay? It's not a big deal to just get your entire genome. It's 3 billion base pairs, 300 copies of The Joy of Cooking for about $1,000. In fact, that's probably cheaper than it would cost you to buy 300 copies of The Joy of Cooking. But personalized medicine is more than DNA. It's going to be the environment too, okay? And we need better tools to study environment. I'll challenge you to think about that. The environment's going to be a big one. But I'll tell you, for those who think quantitatively in the computer world, patient samples data are already there to make an impact across diseases, across medicine. All of these tools, data, services are available on the internet today to actually come up with a new diagnostic biomarker, new therapeutic, and get it trialed. It's amazing. Here's the take-home point I want to challenge you with, all of you. You are all impressive that you spent Sunday afternoon coming to this talk to learn more about the genes, environment, data. The biggest problem is when we go back to see our own doctors, they don't have a clue of what we're talking about, okay? It's up to all of us to teach our own doctors and make them learn about genetics and challenge them, okay? What is it? What is that gene I just read about? And make them go back to the CME classes, because this is the future. That's us as patients now. We need to be demanding this of healthcare. And the only way the healthcare responds is really when patients start to speak up and actually start to demand that this change happens. I have a long list of collaborators that couldn't have done any of this work without a huge list of folks. We'll spend a moment talking about you and Ashley and Steve Quake at cardiology and bioengineering, certainly, and Russ Altman from the pharmacogenomics side. His name is somewhere on this list. I couldn't have done any of this without the generous endowment support from the Lucille Packard Foundation for Children's Health. I was one of the 45 new faculty that came in between 2000 and 2004 based on community involvement and the David Lucille Packard Foundation. You saw that the changes in this hospital. Now we're starting a new initiative to double the hospital size here. I was one of the 45 that came in from the last fundraising effort. My lab started with 0 60 years ago. We have 15 NIH grants now. These 4 institutes of NIH pay me more. These 4 pay me less. Howard Hughes, Hewlett-Packard, Stem Cell Proposition, we work with them. Pharma Foundation, I thank my admin and tech staff wherever I go. I would never get a grant out the door without them. I always thank my wife because she's a biologist. She reads every major paper grant that comes out of my lab. She starts companies with me now, and she generously let me come on a Sunday afternoon to talk to you. So thank you very much.